This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 240, recorded on January 16th, 2013. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiV, the podcast all about viruses. As I'm recording this, it's the week of July 4th, and in the U.S., that's a big holiday. So I've decided to give the TWiV team this week off, but I thought that some listeners might not want to miss a TWiV. So I dipped into my reserve, and I found an episode that I recorded way back in January of this year when I visited the University of Vermont in Burlington. And the weather couldn't be any more different. Back then, it was freezing, and up in Vermont, it was snowing. And today here in New York, it's in the 80s, it's really humid, and it's partly sunny. For that episode, I traveled to Burlington, where I gave a talk in the Department of Microbiology and Immunology. My host was Markus Thali, and I also spoke with Jason Botten, two virologists at the University of Vermont. Markus works on tetraspanins, HIV replication, cell-cell viral spread, and Jason Botten works on arenavirus and hantavirus pathogenicity and immunity. So let's go back in time and check out my conversation with Markus and Jason way back in January. Today is January 16th, 2013, and I'm in Burlington, Vermont, at the University of Vermont. I'm in the Department of Microbiology, and I have found the two virologists here on campus to have a TWiV with. So joining me today, Professor in the department, Markus Thali. Welcome to TWiV.
Hi.
Is this microbiology or is it a bigger name?
It's microbiology and molecular genetics, actually.
Okay. Markus, I met you at a study section a couple of years ago.
Yeah.
And I think shortly after you invited me to come here, and I forced you into doing a TWiV.
Right.
So Markus has influenza.
Soon to be us as well.
Yeah, apparently, we're in a small room. We're in Markus's office. Also joining me today here, another professor from the same department, Jason Botten. Welcome.
Thank you.
And we're in a small room, and Markus is coughing. So these are ideal conditions for viral transmission, right?
Right.
We'll see what happens.
It could be pertussis, though, also.
Pertussis?
Yeah.
Really?
Because they say we have quite a bit, hundreds of cases in Vermont right now.
But these are people who don't get vaccinated, is that right?
No, actually, even with the vaccination, so it fades out over time, apparently.
Yeah.
And in particular, if you're an adult, haven't been vaccinated for 10 years or so, apparently the protection is gone. And
I think we talked about that on This Week in Microbiology. There have been outbreaks of pertussis in immunized people, and they're thinking of going to a second dose, maybe.
Right. Mm. Yeah.
Right? Because they only give one dose at the moment.
Yeah.
Of course, always makes it harder.
Yeah.
I was immunized as a child and never had pertussis. It's a long time ago, though.
Yeah.
Well, maybe I'm still a child. I don't know. Could be. I want to find out where you guys were before you got here. So, Markus, I know you're from Switzerland. You were born and raised in Switzerland?
Exactly.
Whereabouts in Switzerland?
So actually right next to the airport, which is actually one of the reasons why I felt so comfortable coming here, because we have the airport 10 minutes from the office, and so I always loved watching the planes.
What city?
It's called Kloten, so Zurich, pretty much.
Zurich.
Yeah, so at the outskirts of Zurich.
And you went to high school and college in Switzerland?
Yes, high school, and then university, studied molecular biology, got my PhD there.
In whose lab did you do your PhD?
That was Walter Schaffner.
Okay.
Mr. Enhancer. Still Mr. Enhancer.
What did you work on, a viral enhancer at the time?
Actually, I did, even though that was sort of a later project. I mean, I was the lucky guy who I was working on pseudorabies immediately. Yeah. And so at the time we were sort of interested in finding transcription factors that would bind to specific motifs,
right,
and that would determine tissue-specific expression of genes. And so I was looking at that virus, but somehow that just somehow didn't go anywhere. Actually, I started my work with spending maybe a year or so on some factor trap. We were trying to clone transcription factors, and that didn't go anywhere. Then I started doing the pseudorabies immediately, sort of a bread and butter project. But that also sort of took quite a while, and then I was lucky that Drosophila person, Marianne Biens, she had an interest in showing what she thought was the case, that Drosophila homeotic proteins are acting as transcription factors. And because our lab, the Schaffner lab, was one of the labs at that time who had the capability of doing that. And I was running RNA mappings and had everything set up. So at some point, Marianne came and brought us constructs, and then within 2-3 months, I had data together for a nice paper, and I was the lucky guy.
So what made you come to the U.S. for a postdoc?
That was actually viruses.
Viruses. You wanted to do virology.
Well, at the time, it was generally was sort of like if things went well, everybody went to do a postdoc, and most of us went to the U.S. I mean, that's the place to be.
Is the plan eventually to come back to Switzerland, or
it was, yes. I mean, typically people would go to the U.S. and then go back and get some faculty position, or if that wouldn't work out at the time, was SIBA or whatever the big pharmaceutical was sort of the default way to go. So I went and did the postdoc, and I wanted to, so like, as much as I liked doing what I did during my PhD, it was sort of somewhat narrow. You would look at just a stretch of DNA interacting with a few transcription factors, and there was not much completeness, as you may have realized last night. When I'm sort of a big picture guy. I'm interested in big questions, and not so much, like, that's why I would never do structural biology. I mean, I couldn't care less the little details of amino acids, such and such, sticking out there and there. So I'm a big picture guy, and I always felt that viruses maybe give me an entrance into a big picture thing, because you deal with a microbe and cells or host organism, and whatever the virus does, it does it to the entire organism. And so I was thinking that viral systems would be nice to study, and so essentially wanted to widen things.
Whose lab did you do a postdoc in?
I ended up in Joe Sadrowski's lab at Dana-Farber, Harvard Medical, and pretty much the choice was, at the time it was HIV, obviously it was big, there were many labs to choose from, and at the same time it had the advantage of coming to a really big department where everybody was working on the virus. It was absolutely fantastic time that I spent there. Yeah.
And then out of his postdoc you came here, is that right?
Well, no, actually then I did sort of make the mistake to go back
to Switzerland. To
Switzerland, yeah, which was not very productive in the sense that, I mean, we did it because of personal reasons, because our kids, or the older of our 4 kids, were sort of school age, and we thought because of schools we go back. But it's just the Swiss system at the time, and to this point I think, is not ideal for young faculty, because they don't really force you to focus on stuff. So here, I mean, it was, in retrospect, it was completely ridiculous. And I remember actually I showed the proposal that I wrote for the Swiss National Science Foundation at the time to Joe Sadrowski, and he said, so this proposal would feed 10 postdocs for 10 years at least. But they gave me the money, you know, so I went to Switzerland, actually in a very nice place in Lausanne, microbiology, medical microbiology. Heidi Digelmann was the chair there at the time, and so the environment was really great, but it was different because we were a few groups only, and I didn't have all the resources. Yet I had all these ideas of things that I wanted to do, and nobody who stopped me trying these different things. So it literally took me 3 years or so till we finally got the paper out. And so in retrospect, again, I think I would have stayed in the US because I think that would have helped me sort of like in that early independent stage.
So after 3 years, you decided that.
Actually, almost 5 years, 4.5 years later, and because fortunately I was not sort of in a tenure track position, so I knew that at some point I had to choose, and then it was partially my wife, because she, even though she's also Swiss, she always felt like her soul was sort of still in New England or the US. Yeah, I mean, just we had such a good time in Boston, and just you get used to the way people interact here, the positiveness, and so when I was starting looking around, I applied. This was one of two positions. Another one was at Penn State. Penn State was sort of later, and when they offered me this job, then
nice. Once I was in Boston in September at the Needle, this BSL-4 facility, and Paul Dupre is an Irish guy who works there. He's from Belfast. And he was telling me that the Americans have this positivity. No matter what adversity, they always spin it in a positive way. And he says in Europe, this is totally the opposite. When bad things happen in Europe, people get depressed and don't do anything.
Exactly. And I mean, it's just, it really makes a difference. I mean, it's, I wouldn't want to miss it. I mean, I mean, there's lots of positive things, obviously, in Switzerland. I mean, I mean, as you can see, to some extent from whatever the things that I have here. So I'm still very Swiss, like to the
we
clock.
should say that he's got the Swiss railway clock on his wall, which is really cool. I've never seen a small version of that.
Yeah, so that's definitely things that I'm very appreciative of, but ultimately that's one of the things that really brought us back.
So Jason, I remember from last night that you grew up in Minnesota, right? So high school,
college
as well.
I grew up in Montevideo, kind of on the border of South Dakota in the southern part of the state. I went 3 hours north up near Fargo to Moorhead and went to Concordia College.
Wow.
It was almost a complete climate change, just 3 hours by car. And and from there, then I went down to the University of New Mexico in Albuquerque for my PhD.
So were you a biology major in college?
I was.
And you knew you wanted to do research?
Yeah,
college,
I had an outstanding high school biology teacher, Butch Alterman, very unusual guy, and this was back in probably 1990. Took his class, and he had us running DNA out on gels, and it just blew my mind at the time, and so that flipped the switch for me. And the other thing was I knew I wanted to go into biology, and I also didn't like the sight of needles, so I knew I wasn't going to be an MD. And so, but I knew going into college kind of what I wanted to do research.
So mentors are so important, right?
Oh, incredibly important.
They inspire you to do things.
Two people, Butch and then Jerry Woolleader, my track and cross country coach, two of my life mentors, made a huge difference. So how did
track and cross country play into your goals?
I think working with Jerry, that guy was just... You talked about mentors this morning. He was the guy that got the best out of everybody without... He didn't have a whip. He didn't raise a hand. He didn't raise his voice. The guy just silently and very effectively got the best out of you. And
did he make you feel confident?
Oh, absolutely.
Yeah, that's important. That translates to
and that what we were doing was incredibly important, and that we were doing it well. And
I think kids need that so much.
Yeah.
Right.
Yeah.
And actually, speaking of making you feel comfortable, and that was one of the really nice things of my mentor, Walter Schaffner, because from the beginning, I mean, he encouraged us to get in contact with our competitors and friends in the US. Like we would communicate with Tashney Lab and Maniatis or so directly, and it was not him. It was everything went through us. I mean, he encouraged us to do that, and so, yeah.
So you went to New Mexico for a PhD.
Yeah.
You told me a story last night about how you ended up in a lab there.
Yeah. So I went down early because I had this idea that I was going to help cure cancer. I was going to work on DNA repair pathways, and I figured what an opportunity, because UNM had an appointment with the Los Alamos scientists, right? These world-class guys studying this. And I was going to go up and do a rotation up there, so I went 2 months early before the year started, and I got there, and the guy I was going to work with didn't get a clearance. I needed a government clearance, and why don't you come in the spring, he says. And so in the meantime, I had this other guy that I had interviewed with, Dr. Brian Hella, who worked on hantaviruses, and I just thought it was such an interesting topic he was working at. All right, and he said, you can come tomorrow and work for me. And so I went into his lab, and I actually never left.
So was this after the Four Corners Hanta outbreak?
Yeah, so the original hantavirus outbreak was in 1993, and this was in the summer of 1996. So this was still very fresh in the memories of people around there and in the country. And I walked in, and literally, my first summer, I had a very multidisciplinary training experience there. It was very unusual, and it's one of these things where you apply to all these schools, and where do you pick? And I'm a big believer that there's opportunities wherever you go. But I worked with Brian, who was he's an MD who did only research, so he wasn't seeing patients or anything. So he was a molecular virologist, and I also worked with Terry Yates and his group at the Museum of Southwestern Biology. And Terry, for people that knew him, he passed away a few years ago, unfortunately. He's a world-class mammologist, and so my project as a graduate student was to do three things. One was to build an outdoor biosafety level 4 lab so that we could use this new facility to safely take deer mice, which are the carrier for hantaviruses, the reservoir, and start an animal infection model using those animals.
What do you mean by outdoor? Just you built a building, or it's just open air?
No, it's basically. Well, so here's the conundrum, right? So if you work on hantaviruses in the Americas, they're considered level 4 if they're in a mouse, because the animal can excrete virus, and then it can be transmitted through an aerosol, and it has a 40% fatality rate, and so you put all that together, and that's pretty scary. But the CDC published these guidelines. So Jim Mills and his colleagues put out these guidelines saying that if you were to go outside and trap wild mice that have these hantaviruses, as long as you're wearing a Tyvek suit and a powered air respirator and gloves and booties, because you're outdoors and the material that's shed is going to be dispersed in an ocean of air and the UV can act on it, that's acceptable. So what we did, in essence, was we took a square section of land in the middle of the Sevietta National Wildlife Refuge, right? So this is 250 hectare place that's all fenced off and patrolled by the Fish and Wildlife. We went right in the center of this thing, found a square area of land, and built a fence around this, right? The fence went deep underground and high above ground so that any rodents we put inside that couldn't get out, and anything outside couldn't get in.
So it was a wood fence? Or
no, it was metal. It was
metal.
actually
It's not chain link, right?
No. It was solid. And then inside that fenced area, that was our working area, and there was no roof, right? So we had the sunlight and we had the open air. And then you showed a picture this morning how Peter Palese said, well, if you need shelves, you need to build them. So when I was an undergrad in the summers, I poured cement, and so I got really good at wheelbarrowing. And
why pouring cement? Is that for the fence?
No. Well, that was just to make money as an undergrad, right?
Okay.
To get through. So then, as a graduate student, Brian said, "We need to build this thing. Are you good with tools and with your hands?" And I said, "Well, I've been doing this."
Yeah.
So anyways, I rented a bobcat and we dug the ditches, and then the complicated part was then we had to build cages inside the facility so that we could house the animals. And it gets, it'll get down to 10 degrees Fahrenheit at night in the winters out there, and it'll get above 100, 105 in the summers.
Yeah.
And so anyway, so we went around and settled on a nest box, which is basically we would dig a big hole and we'd bury a 30-gallon trash can in that.
Right.
Right? And you'd take the lid and we'd cut the center of the lid out and cover it with mesh. So now you'd have this basically enclosed thing that's buried in the ground. And then inside the 30-gallon trash can, we took 5-gallon paint pails. And we constructed, it's hard to describe without seeing them, these artificial burrows that we could put inside the trash can, surround it with dirt, so that these animals could have a little safe area to live and do their thing in. And
this is within the fence, right?
all within the fenced area. And so we got that working. We ran some different species that we trapped on the refuge through, and in the end, I worked in that thing for about 5 years through all weather extremes, and it worked like a charm. So then, and then the next step, we'd go up and literally out on the refuge trap for deer mice, so that we could find, because you can't buy these.
Right.
So we found exotic deer mice on the refuge, put them in, and we had to quarantine them then. The first thing was to make sure that they hadn't recently acquired a hantavirus infection, so we'd bleed them and make sure they didn't have antibodies, hadn't seroconverted, give them 6 weeks, make sure they didn't seroconvert at that point, and one of them did, showing the importance of doing that.
And this time they're in your artificial...
Yeah, each mouse, one to a cage so that they're all isolated, and then moved them up to campus and got a wild-caught colony going. And then the one mouse that seroconverted, we took the tissues from this female, and ground them up and got this to work, which was kind of phenomenal at the time, because my first meeting I ever went to was going to the International Hantavirus Symposium, and it was in Atlanta, and so CDC played a role in hosting it, and I met some people there, including Jamie Childs, and he said, "Tell me about the project you're going to work on." I said, "Oh, we're going to build this facility, and we're going to set this model up, and we're going to study transmission and the natural history." And he said, "Oh, that's really great." He said, "But it'll never work. It'll never work." He said, "We've been trying to do this for years here, and we can't get this to work." And I think in the end, what worked for us was we were using not only mice that were the right reservoir, but we were using the virus from that population that we founded the colony with, whereas they were using some mice from the Peromyscus Stock Center in South Carolina. They were originally derived from Michigan animals, and then they were using lung homogenous from New Mexico samples. And the hantaviruses are really finicky to grow. I mean, they have a really restricted host range, and so...
So these mice in your outdoor BSL-4, the main point of that was to get them then in the lab and make a colony.
Yeah.
And then did you do transmission in the lab then?
No, so then
it was all outside?
It was all back down at the
So you would breed them inside and then put them back out at the site?
Yeah.
Is that how? Because you would want them to be outside in sort of a semi-natural environment.
Yeah.
And the virus you used was one you had originally gotten from that very site, from mice in that site, right?
And so the beauty of that was if a mouse had escaped, it was 20 home ranges away from the nearest people, because it was way in the middle of this big refuge.
Got it.
And two, if it got out, it would be bringing virus that was already there in the first place. So neither the mouse nor the virus were exotic to that area.
So if I want to trap mice in New Jersey, do I have to wear a Tyvek suit and a powered respirator?
Well,
I should, right?
You should.
No one.
If you were going to follow the guidelines, you would.
Yeah.
Now, if you go out and spend a month in the life of a mammologist, if you hang out with one of these crews that go out, and these are fascinating, really interesting and likable people. I liken it to the construction work sites that I used to be on. That there's a real precedent for prior to '93. Nobody knew that deer mice carried hantaviruses. Nobody wore protective gear.
Yeah.
And Terry Yates used to have a picture he would show. It was really striking. The first picture he'd put up on the screen was pre-1993. And you'd have about 6 people crowded around a picnic table, and they've got bottles of ether, and they've got capillary tubes for orbital bleeds, and one guy's bleeding a mouse while someone's feeding him a bite of a sandwich, and the person across from that person's drinking a beer, and all of this is going on, and nobody's wearing gloves or anything. It's just. Yeah. And then he shows post 1993, and they're all suited up, and it almost looks like they're working in a site on the moon, because they're all sterile and they're in these. Yeah.
So the hanta that you got out of this mice, Peromyscus specifically, right? Peromyscus maniculatus.
You got it.
It's a common mouse in the U.S., is that right?
It's actually the most abundant mammal in North America, that species.
So I'll find it in New Jersey as well. Right?
Yeah.
So that virus, would that be able to cause a human infection? So
absolutely.
So the virus that caused the outbreak in the Four Corners, that came from a mouse, but did that spread among people, or were they all primary infections from different mice? Yeah,
that's a good question, especially with the high mortality rate. No, the most of the infections are all of them are thought to be mouse to human.
Yeah. So these recent ones in Yosemite, I guess, those were all brand new. So it's a mouse virus that just jumps into people.
Yeah, and the typical exposure, right? If there's one thing that the audience can learn today, it's how not to get hantavirus infection.
Okay.
Because it's common, right? We've all had mice that get into our homes or our garages or our vacation places. And so, I just taught this virology course, and I said, if there's one thing you guys learn, it's got to be this, right? Because Anyways, so if you walk in to your basement and you see you've got droppings, I mean, typically you'll see the droppings, you'll see dried urine on the floor, and then all the nesting material. It's got both of those things in it. So the virus is thought to be excreted in urine, in feces, potentially in saliva. So the last thing you want to do is grab a broom or a vacuum, because that's going to aerosolize this thing. And what's dangerous in North America is exposures indoors, because you don't have a lot of air circulation. There is no UV acting on anything that's been shed, and most infections have been linked to indoor exposures. So the deal is, you go into that room, and if there are windows, you open them and leave and give it an hour or two to air out and let sunshine come in. And then you go back in and you spray everything down with bleach, 10% bleach, and give it 30 minutes to just soak up and absorb. And then you go back in with gloves and bleach-soaked towels, and you wipe it up carefully into Ziploc baggies and then toss it.
So the first recognized hantas in the U.S. were at the Four Corners, is that right? Since then, how many infections do we know of?
Well, it's in the hundreds. I should know the exact number. It's probably in the 400 or 500 range, somewhere in there.
Mainly in the Southwest?
Most of the infections are on the western half. Like if you look at a map on the CDC website, it'll show that most of the infections have from the middle up to the western half of the side of the country.
There have just been a few in the Northeast, for example, right?
Right.
I think there was one in upstate New York recently.
In the Adirondacks.
Right.
Yeah,
and
I think so.
There was another one on Long Island years ago.
Yeah, well, that's New York virus, right? That happened on Shelter Island.
So that's a different strain of hanta.
Yeah, it's very very similar to Sonombre. It's carried by the white-footed mouse, Paramyscus leucopus. In these Paramyscus species, it took me 6 months to be able to tell a white-footed mouse from a deer mouse.
Okay.
You got to measure their ears and their tails, but.
Had there been hanta infections elsewhere before? I seem to recall in Korea that's where they were.
Yeah, so
right?
they've been documented for hundreds of years actually, but it came to Western medicine's attention during the Korean conflict. There was about I think 2,500 U.S. troops that came down. The disease over there is called hemorrhagic fever with renal syndrome. So it's mainly kidney centered, and over here in the Americas it's called hantavirus cardiopulmonary syndrome, and so. You get capillary leak and things like that in both settings, and shock is a big contributor to death. But
was it as deadly as here?
No, it depends on the species. So hantan virus is the one that was originally identified in Korea, or over in Asia, I should say, and along the Hantan River Valley, and that one can have a mortality rate of like 1 to 10%. You have Sol virus carried by Rattus and Rattus norvegicus, and others, and you also have pumalavirus that's in Europe. So the European is probably maybe the least lethal blend of maybe 0.1%. But if you had to contrast old and new world, in the Americas you have very few cases, very high mortality rates. In the old world you can have up to hundreds of thousands of cases per year, so high morbidity. So they're definitely a significant problem.
And rodent reservoirs in all cases?
Yes. Yep.
But different species of rodents?
Yeah, absolutely.
And we don't understand why North American cases are high mortality, right? No,
not really.
And
the other interesting thing, I think, is, I studied transmission as a graduate student, and, so we did experiments where we deliberately infected mice, and we would put these index mice, they were infected now, we'd put them in a cage, and then we'd add in a couple of uninfected mice with them to see could they transmit virus over their first week of that infection, and then we'd swap in new ones and see are they infectious the second week. So out of 59 mice that were exposed to acutely infected index mice, only one got infected during those experiments. We collected urine, feces, and saliva, and we couldn't detect infectious virus in any of the samples, and so but I think what it reflects is we still have an incomplete understanding of how the virus is even maintained in nature. We think it's horizontal. Mainly the males get it in the wild populations. But how they transmit it, is it through aggressive behavior? That might be a clue. Is it through... we just don't know.
Yeah. And the mice are fine with this virus, right?
Yeah, you wouldn't know.
How long do they produce virus? Is it lifetime or a short period?
Yeah, so that was kind of interesting. That was one of the questions we had when I was a graduate student. Are they going to were they going to clear this? Will there be a disease model? And we pulled 20 tissues from the animals. We could see no histopathology as a result of infection. And actually, it's one of the slower growing viruses.
Yeah.
So you can barely detect RNA by nested RT-PCR in tissues only beginning at day 7. So unlike a lot of these viruses we study, by day 3 or 4, they're hit their peak and they're on their way down. And these actually will have peak titers around day 21. And then the animals they remain persistently infected. Yeah, and we noticed 2 different phenotypes, and these viruses are very cyclical in terms of their patterns of quiescence and recrudescence, so we don't quite understand that. The arenaviruses are like this as well, other rodent-borne viruses. But we noticed 2 distinct classes. We had mice that they would look acutely infected. They'd be viremic. They'd have virus in almost every tissue we'd look at, a panel of 10 or 12 tissues. You'd have other animals that would be completely restricted to the heart, or the lung and the brown adipose tissue as well. And in those cases, they didn't have any replicative species in those tissues. It was just the genomic RNA and the nucleocapsid that binds that. And presumably, polymerase would be there too. And so one of the interesting hypotheses might be that these viruses can actually go into periods of quiescence where they just maintain themselves in a certain strand-specific manner. And we were interested in what signals might reactivate the virus.
We just did a paper on TWiV by Matthias Schnell, rabies.
Yeah.
And they have a mouse model they infect. And they find rabies RNA with nucleocapsid protein bound to it for long periods without replication.
Yeah.
And the nucleocapsid protects the genome so it doesn't degrade. And there's no evidence of replication at all. So maybe a lot of these viruses can do that.
Yeah, and we're addressing that now. One of my graduate students, Ben King, has developed a really beautiful fish assay to look at genome and antigenome and the mRNA species of arenaviruses. And because if you put them into culture, they'll get this 48-hour spike of 10 to the 7th PFU per mil, and then it'll come down by day 7 to just about maybe 2 logs, and then it goes through again this very cyclical progression, and you can set your watch by it. And so he's looking now to see whether different species of the genome and antigenome are maintained over these periods and how it correlates.
Right.
And,
so what fraction of wild mice are seropositive for hantaviruses? You have any idea?
Any given site, you'd have anywhere from 5 to 40%.
Varies per site, right?
Yeah, and by year or conditions.
Okay. So you came, after you finished this postdoc, this PhD program with Brian, you came right here to Vermont, is that right?
No, then I went out, my next stop was even further to the southwest. I went to San Diego next, De La Hoya, and I worked with Michael Buckmeyer at the Scripps Research Institute. So I went from doing the rodent ecology and transmission to switching to arenaviruses. So again, rodent-borne viruses, very, very nasty. Loss of fever would be an example. Bolivian hemorrhagic fever or Argentine hemorrhagic fever. And I decided to be a good virologist, I had to understand the host response to the viruses, so I wanted to get into immunology. And really what I ended up working on there, and I worked with 3 people for mentors. So Michael, Alex Sete, who was at La Jolla Institute for Allergy and Immunology, and then Lindsay Witten at Scripps. So that was kind of my 3-pronged mentorship at the next stop. And by and large, we worked on mapping human T-cell epitopes from the pathogenic arenaviruses and then using them to make multivalent vaccines consisting of different combinations of epitopes so that we could generate multivalent vaccines. And so we used HLA transgenic mice to use them as a human platform or a model for testing. And so do we know that T-cell responses are important for a vaccine against these viruses? Is that Yeah. I mean, I think the idea of finding conserved elements like T-cell epitopes or other areas of the antigens of the viruses as a multivalent approach is relevant. Like flu, can you get around antigenic seasonal variation by going to conserved regions? But in the case of the arenaviruses, we know that certainly LCMV has been the model that helped define MHC restriction. You get one of the most potent and florid T-cell responses anywhere in nature if you look at LCMV in the mouse. So in humans, for the old world viruses, LCMV and Lassa, people don't make neutralizing antibodies. And so it really is T-cell mediated,
response.
and that's been worked out in the mice
Yeah.
as well. And so there it would be one case where it would make sense. You would really want to tailor your vaccine to be T-cell specific, and so
Right.
that was kind of the rationale for that.
And then you came here from Scripps?
Yeah.
How long have you been here now?
4 years now.
Pretty new, right?
Yep. And and I've reinvented myself again, for better or worse. And now I'm mainly interested in so the viruses we study are hantaviruses and arenaviruses. They only have 4 proteins, right? And we know virtually nothing about what those 4 proteins are doing in terms of interactions with host cell proteins. And so we've been taking a large view of, first of all, what is the global interactome between these small number of proteins with cellular proteins, and then interrogating those interactions to see which ones beneficial for the virus, don't matter, or otherwise, things that may contribute to pathogenesis. And so I've got 3 really talented students. Joe Klaus is the guy that we could call glycoprotein because he's working on the glycoprotein. Ben King works on the nucleoprotein, and Chris Ziegler works on the matrix. And these guys have all done these beautiful jobs of coming up with their proteomic screens. And so we've got a really good idea of the interactome now. And we're moving on then to find specific targets. And the idea is, at the end of the day, hopefully we're going to advance our understanding of how these viruses are interacting with the cell. And at the same time, if we find important cellular proteins that are required for the life cycle or required for pathogenesis, we have targets for antivirals and therapeutics. So that's kind of the program we have
So
now.
we had glycoprotein, matrix, nucleoprotein, and what's the 4th? It must be
the polymerase,
right?
and we've left that aside for now because it's a big unwieldy protein.
And the genome is negative stranded.
It is.
And how big is it?
They're bisegmented. The L segment is about 7.2 kb, encodes the large polymerase and then the small matrix protein, which is the budding factor. And then the S segment encodes the nucleocapsid, or the nucleoprotein, which
this is the hanta or arena?
This is the arena now.
Right.
Yeah, and then it also encodes the glycoprotein.
Okay.
Yeah.
So the interactome study, where you look for viral proteins that interact with host proteins, and you do this in a massive way, so you get all the interactions. You've done that also with hantas, is that right?
We've done we're starting to do a little bit.
So how many proteins do they encode for also?
Yes, they do. The hantas are very similar. They have their own polymerase and glycoprotein, and in this case, nucleocapsid. And then they don't have a matrix that we know of, and that's still kind of out there as to how their budding is probably something to do with their glycoproteins. And they also have a small protein called NSS, which stands for the non-structural protein of the S segment. And the only known function that I know of is as a type 1 interferon antagonist. In fact, both of these viruses, like 3/4 of their proteins are devoted, part of their function is to be antagonistic to type 1
interferon is a great antiviral system.
Yeah, and they're very sensitive, obviously.
And so viruses have to. The ones that don't encode antagonists don't exist anymore, I think, anyway.
Yeah. Yeah, and so to give you an example of what we're really excited about now is we found a protein called ERGIC53, which stands for the ER-Golgi intermediate compartment protein of 53 kilodaltons. And so if you can picture the secretory pathway in your head, you have the ER, you have the Golgi, and for proteins to get from the ER to the Golgi, they have to travel through the ERGIC, this intermediate compartment between the two, and this protein's prominent between the two. So in our glycoprotein screen, this came down, and I started to read about it, and it's an intracellular cargo receptor. So it only has 4 ligands that it's known to be important for moving within this pathway. So if you lose ERGIC53, they no longer, these cargo no longer traffic from the ER to the Golgi, and the 2 prominent ones are coagulation factors 5 and 8. So there's people walking around all over that are null for ERGIC53, and they have really mild bleeding symptoms, but they have deficiencies in factor 5 and factor 8. And so you have all these proteins, all these candidates. One of the things that drew us to this was that not only does it interact with hantavirus glycoproteins, it does with arenavirus glycoproteins too. It's highly conserved across all the pathogenic members. So we thought, all right, so here you have human infections where in both cases you have deficiencies of factor 5 and factor 8 reported in the literature, so they have bleeding abnormalities. So maybe our glycoprotein interacts with ERGIC53, disrupts ERGIC53's ability to move these factors, and maybe that's why people bleed during infection. But more importantly, if ERGIC-53 is a cargo receptor required to move things through the secretory pathway, maybe the glycoprotein maturation depends on this protein to move. And so we've tackled the latter part to see if it's important. And in fact, it's highly conserved in its importance. It's a dependency factor for old and new world arenaviruses. And it's kind of cool. So we've been able to show that if you infect, ERGIC-53 normally doesn't go beyond the Golgi, or even the cis-Golgi for that matter. But during infection, this thing goes right up to the plasma membrane, and it goes right to viral budding sites. So we have beautiful super resolution images showing that, and it gets into the particle. So the bottom line, though, just to sum it up, would be that in this case, if you restrict ERGIC-53 from leaving the ER, so if it's stuck in the ER, or if you just get rid of it altogether in null cells from humans, you make viral particles. The glycoprotein still traffics, which is surprising. It goes to the surface. You get these particles, but they're no longer infectious. And the main difference that we've got at this point is one class of particles that's infective have ERGIC-53, the other do not.
Yeah. So these are cells from people who lack ERGIC-53 you can use for, and they make non-infectious virus particles.
That's right.
And that's what I was going to ask you.
Yeah. So, and it may suggest that there may have been some selection along the way. People exposed to rodent-borne viruses may have helped select for this particular mutation in the population.
Sure.
So anyways, it's one example of, you have all these proteins and you have to pick, and I think we were lucky we picked on a good one to start with.
So you don't trap mice anymore?
Only at home. In fact, when I moved here, I was out mowing my lawn. We have these voles that get into our yard here in Vermont. And I was out mowing my lawn, and I was watching these two. They were running along my house, but over by the garbage cans. And I jumped off my new John Deere mower and caught one of these two mice. And I picked it up, and I got that little, SOB. And then I looked over, and there's my neighbor, and his jaw was down on the floor. He couldn't believe that he just watched me going. So anyways, but all the years in New Mexico chasing wild mice, they're very different from the inbred strains we see in the lab. They bite, they jump, and they're really quick.
But do you have Peromyscus colony here?
I don't.
You don't. So you don't do any mouse work any longer?
No, I've kind of left that behind for now. Although it looks like we may get back into it because there is an ERGIC-53 null mouse that we may have to look into here. So
a lab mouse can be infected with hantaviruses?
You can. I've mainly used lab mice for the arenaviruses using LCMV. For the hantaviruses, I was just talking with Joe Prescott, another hantavirus researcher, and I think he's got data showing that he can put, I think, Sonombre and Andes virus, one of the South American hantaviruses, into lab strains. They don't get disease, but by and large, you're restricted to usually the reservoir rodents. It's really tricky, so you have to. And then there are no reagents. So
you're a relatively new investigator here, right?
Yeah.
So a lot of people listening who are considering careers in science. So can you give them some advice? Like what's been the tough part for you to get going here? Or is it all easy?
Oh no, I wouldn't say that it's all easy. My advice would be, when I was looking around to try and find. So my path when I was at Scripps, and I kind of made a deal with the devil, with our chair, if I can get a grant, will you promote us? So a couple of us got promoted that way. But I always knew that wasn't kind of the last stop for me. And then when we had a child, my wife put her finger on Boston on a map and said, you have a 3-hour driving radius, make it happen. And so
their family's in Boston.
Yeah, she's from Boston. Yeah. So we knew it was going to be there in this area. And so I don't know, when I was looking around, what drew me to UVM was it was a little smaller, but through the COBRA program, where you can get funding for some of the smaller states that don't traditionally get as much NIH funding, really strong cores were here. So it even though it was small and very intimate, you could do a lot of high-powered science here. And what drew me here was the kind of almost the family feel and the highly collaborative, just the feeling I got, and it hasn't changed since I've got it. I really have great mentors, like Markus is one of my faculty mentors here. So I think, I don't know if I'll ever reach a point in my career where I don't need mentorship or help from others. I don't think I'll ever reach that point, but I think it's really important that you land in some place that not only has the facilities and the equipment you need, but you have to have the people. Without the people, it's really difficult.
I still have mentors.
Yeah, I mean,
Yeah. I've senior people in my department who were there before I came. They still help me. I go to them for advice, for sure.
Absolutely, yeah.
So are you the only two virologists here
Pretty much.
on campus? Yeah.
Yeah.
I know Sally Huber is a viral immunologist, right?
Right.
She's really interested in the
Yeah.
immune side
response,
more.
right.
So you guys talk a lot? Do you have lab meetings together or some kind of...
Indeed, we do, yeah. So, and actually that was really nice for me because I remember vividly that when Jason came here, originally I was a bit nervous because he was doing the T-cell epitope things, and thought, I don't really want to have a viral immunologist, you know. So, and it was really great, and that it just so happens that actually I think it was for financial reasons, because I think you had money to do the T-cell epitope, but you couldn't really take the money here, if I remember correctly. So partly for that reason, you started really getting into cellular virology, and that was just perfect. And I think, I mean, that was actually something that people sometimes ask me, well, why did I go here when there was nobody working with HIV or so? But I keep saying that for me, interacting, for example, with Alan Howe, he's a cell biologist here, in some ways is more important because, I mean, viruses are part of the cell. So I, in some ways, learn more from talking with cell biologists than working from people working on HIV or so.
Yeah.
And so I think, I mean, but still, it's nice to have now two groups, and we have started doing joint lab meetings, and it's nice to see that the students certainly can profit from each other's presence and expertise, and particularly also when it comes to whatever medium we are big into, imaging or so, and so I think
there's just enough. We're divergent enough, but there's enough overlap that it really works well.
Yeah.
And I think it's I used to have a Monday morning meeting with just my group, and now we've shifted to the every other week with Markus, and that seems to work really well.
My last question for you is, funding is tough now. It's tough being science. What do you think? Have you felt that, or do you see it's not an issue for you?
Oh no, I've certainly felt it.
How about your people, your students and postdocs? How do they view it?
You know, I think it's a really hard time right now. I mean, when I was looking to take a faculty job, everybody said to me, "Oh, this will be great. You'll get a startup package, and by the time that runs down, everything will be good again." Well, everything is worse than it was when I took the job originally. So, I mean, we've had good fortune to catch some grants along the way here, and we're battling to get another R01 or get one. So, but I think for the students, it's, in my lab, it's a mix. We have one person who he knows what he wants to do. He wants to go out and teach, and he's going to be a great one. And the other two, I think they would like to have a PI position somewhere, and I think it scares the what out of them to kind of watch the frantic things and the efforts that are going on to try and get funding. And so I don't know, I hope things turn around, but for now I'm not giving up the fight, that's for sure.
This is what you want to do, right?
Yeah, I love it.
So Markus, I want to talk a little bit about what you do, and I wanted to tell you that I was reading on your webpage here and noticed tetraspanins is very prominent. And we just did it this week in parasitism. Yep. Schistosomes have a tetraspanin on their surface, and this is being investigated as a vaccine target. It also seems to be an immune defense protein, somehow shielding the schistosome. And on the episode that we discussed this, we had a postdoc from Steve Goff's lab join us, because my co-host Dick Despommier got him very interested in this, and he wants to do some experiments now to try and put them into mammalian cells and see if they can shield or something. So, what do you do with tetraspanins in mammalian cells?
All kinds of things. I mean, I should say that we developed this interest in tetraspanins.
That's just part of your interest, right?
Right, but it's become pretty much a major part of our interest. I mean, the last the one that still funds us right now, even though we don't use the word tetraspanin in the title, or it's pretty much about what tetraspanids do for or against HIV. And started out relatively harmlessly, because we were looking to use tetraspanins as markers we were interested in studying release mechanisms, essentially.
Virus release.
You're right. Not necessarily the escort ones, but just more spatially how the virus is released at viral synapses, as we know now, and the regulation of these processes. And tetraspanins turned out to be, actually, I should say, the first time I tested if tetraspanins do something to the virus was when I was still in Lausanne, Switzerland, because at the time, John Elder at Scripps, he published a very nice study showing that a particular antibody against CD9, one of the tetraspanins, inhibits release of feline immunodeficiency virus. And then, sure enough, at the time I got in contact with Eric Rubinstein in Paris. He's one of the biggies he had all the tools and antibodies against tetraspanins. So we got a bunch of antibodies. Turned out that none of them did do anything to HIV. And then for a while we sort of abandoned things, but then again came back because after all, what has been shown very early on, in the early 1990s actually, is that tetraspanin, in particular CD63, is specifically incorporated into HIV. And that always I found that puzzling, because CD63 turns out to be a resident of late endosomes, lysosomes. And I always thought, well, that's sort of astonishing. Why would late endosomal protein be enriched in viruses that but mainly from the plasma membrane?
So this CD63 is not on the plasma membrane.
Right. I mean, it's like less than 5% of a total population is there. And actually, the nice work by Juan Bonifacino, for example, shows that it goes there on its way to late endosomes. So it recycles essentially from the surface. And so I thought this could be an interesting marker to define the exact exit sites, because if CD63 gets specifically incorporated into viruses, it means that it goes to the sites where the virus is released. And so that's pretty much how we got into tetraspanins again, and then we published a paper on that in 2006, where we showed that indeed HIV buds through microdomains at the plasma membrane that are enriched in tetraspanins, actually not only 63, but also CD9, 81, and 82 at the time. That's what we showed.
Is 63 required for release?
No, unfortunately.
But 9 is, right?
Well, also not. So this is actually, sure enough. As I mentioned, we were mainly using tetraspanins as sort of markers for release, because we just thought we need to get a better idea of where the virus is released. But then, as I mentioned, I think yesterday quickly, when I tried to renew my grant, study section members didn't really like us sort of just doing this descriptive science, showing where the virus is released, but they wanted to have some function. They were asking exactly the question that you asked. What does it do for the virus? Is it involved in the release? But we knew already from the experiments that we did in Switzerland that likely it would not be involved in release, at least it's 63 based on the antibody work. But there was still the data on the CD9 antibody that inhibit FIV release. And so I got in contact again I got in contact with John Elder and asked him for the antibody. And for a while we actually did then parallel experiments with FIV, and we tried to get our hands also on a human anti-CD antibody. And it turns out that one of them, K41, actually does inhibit release. But which was nice at first sight, but turned out to be actually not so nice, because it's not what this antibody does. It's very peculiar antibody, and what it does, it essentially clusters CD9 and actually also other tetraspanins, brings them together and probably just physically somehow screws up the release, the exit of the virus. So it's likely not a specific mechanism, but it's an antibody-specific thing. So there's still some controversy. I mean, there's one group in France. They have published that if they knock down one of the antibodies, one of the tetraspanins, CD81, they actually get some inhibition of release in some T cells. But in our hands, or also Mark Marsh at MRC, they haven't seen these effects. So in our hands, we don't really see a contribution to release. What we have started then really focusing on is, and that's really what tetraspanins are well known for, is they are involved in regulating fusion processes, and the most prominent one is oocyte sperm fusion. So where CD9 was shown in the late 1990s to be absolutely required for fertility of mice. So that was shown in mice. So CD9 is absolutely essential for fusion of sperm with egg, and it needs to be expressed in eggs. There's other fusion functions also that are regulated, like formation of giant macrophages through fusion of different macrophages is regulated. In that case, that's negatively regulated, likely by tetraspanins. So tetraspanins naturally seem to regulate fusion events, and so we were looking at that. And coincidentally, a group in Japan actually also was looking at that, and they showed half a year before us that overexpression of tetraspanin makes virus less fusogenic. And we got the same results and sort of were puzzled because so we know since 20 years that the virus incorporates CD63, and yet now we show that if it incorporates that, it makes it actually less fusogenic. So why would the virus do that? And so from that we were looking at cell-cell fusion, started looking at cell-cell fusion, because as mentioned before, I mean, and as probably most of the listeners know, HIV is supposed or is thought to be transmitted primarily at so-called virological synapses, really when producer cells and target cells align closely. That's the most efficient way for a virus to be transmitted. And actually that goes back also 20 years ago. It was already shown that this way of transmission is very efficient. And so we were thinking, if that's and that's actually something that I discussed, I remember with Heinrich Göttlinger, for example, in the late 1990s, we were always puzzled. I mean, if you think about the situation, you have a producer cell that obviously expresses envelope glycoprotein on the surface, and getting in touch or in contact with a target cell, which expresses the receptors, the core receptors. Why don't they fuse when they form a synapse? And we know, and everybody who studies HIV knows, I mean, like you bring producer and target cells together, they can align, they stay together, kiss each other for like 20 minutes or so, and then they separate again. So why would they not fuse?
Because paramyxoviruses in the same situation would fuse, right?
Yeah, right.
Neighboring cells form syncytia.
Yeah. And I mean, every once in a while you see syncytia forming, but for the majority of cases they don't. So they can align and not separate. And so we're starting to think that perhaps that's really why the virus evolved to recruit tetraspanins, to prevent the fusion of producer and target cells at the virological synapse, while allowing the virus to be transmitted, to be released in the synaptic cleft, and sort of it's like a compromise. That's how we look at it. I mean, the virus agrees to incorporate some of these tetraspanins, become a little bit less fusogenic, but hey, it's in the synaptic cleft, it's right next to the target cell. Even if it's a little bit less fusogenic, it will still make it into the target cell. Yet the synapse can form and can disengage again. The cells can disengage again. So that's pretty much the focus of quite a bit of what we're doing right now. So we're trying to find out the mechanism with which tetraspanins inhibit the fusion, and that's pretty. I mean, it has brought us, and actually brought me quite a bit into the tetraspanin community. It was nice when we published this paper in 2006. I was contacted by the organizers of that year's FASEP conference on tetraspanins, and they invited me to present the stuff. And then, sure enough, I went there. And then 2 years later, I was invited again. I chaired a session on infectious disease because it turns out, I mean, you mentioned the parasites, but obviously there's very prominent viruses, HCV, that needs CD81. So there's actually quite some microbes out there that use tetraspanins either positively or negatively for their replication. So I got quite a bit into the community of tetraspanins, and was really great.
So you mentioned lipid rafts before. So are all the tetraspanins in lipid rafts? Why don't you explain what a lipid raft is in case someone doesn't know?
Okay, so, well, it's a tricky issue, you know. Well, I'm not a raftologist, but I guess unfortunately the concept of lipid rafts has been screwed up quite a bit for years and years, and people. I mean, I think what the idea that people had for a long time was that they're sort of pre-existing platforms, and even though the original proponents, Kai Simons and others of the raft hypothesis, they always said that no, they're transient assemblies of lipids or so. I think that was not really clear, and people were still sort of had that idea of pre-assembled platforms, and so with tetraspanins, so the relationship is like if you do the classical fractionations or so, tetraspanins partially co-fractionate with raft components, but only partially. So they have always been sort of separated, and so they're a different entity. But there's partial overlap, and certainly with regard to HIV release, I mean, we know that the virus goes through domains that are enriched in lipid raft components, and as I mentioned before, also,
so it exits cells through lipid rafts.
Right, but the thing is, it only forms those rafts. So again, the concept of the raft is sort of, I think, misunderstood by many people. It's not that the virus chooses to go through a whatever 200 nanometer wide raft, but when it assembles at the plasma membrane, when GAG multimerizes, then those mini rafts, which are whatever nanometers or so wide, they are trapped there. And actually, 2 years ago, we showed this very same thing for tetraspanins also. So we originally thought tetraspanin were pre-existing platforms through which the virus would pass. But then we showed with FRAP analysis, again, about 2 years ago, so that just like raft components, they are not pre-existing platforms. They are assembled only when the virus multimerizes, the viral proteins. Yeah.
So your idea is that the tetraspanins are involved in production of particles as well, or mainly?
I would say in the safe transfer of particles to target cells, and they're acting actually on both sides. A little while before we published our studies on their role to prevent fusion in the producer cell, Francisco Sanchez-Madrid in Madrid showed the same thing for the target cell side. So he showed that tetraspanins on the target cell side also prevent fusion from taking place. So they are really sort of like controlling elements on both sides of the synapse.
So it prevents cell-cell fusion.
Yes.
But when the virus has to infect the cell, does it fuse at the surface, or is it taken up by endocytosis?
That's still controversial.
Yeah.
So, as you may know, there's very strong opinions on either side of that. So, I mean, until a few years ago or so, the opinion was that there's all fusion happens at the plasma membrane and there's no endocytosis. Then there were a few papers by Hans-Georg Kreuslich in Heidelberg that show the involvement of dynamine and the uptake or role. And then obviously there was the cell paper, blanking right now, 3 years ago, I think, that showed evidence for uptake through endocytosis. But there's still controversy to some extent, because turns out, and actually there was just a very recent nice review by Bob Blumenthal, one of the foremost experts in fusions, and where he points out that some of the phenomena could be explained by, for example, the fact that dynamine actually plays a role, probably also in fusion. So it's not necessarily that you have to go for endocytosis. So, but yeah, that's about the whole idea.
Okay. So if the particles are say taken up by endocytosis, then the inhibition of fusion from cell to cell by the tetraspanins, that has to be overcome even in the endosome, right?
Say it again.
So normally tetraspanins prevent an HIV-infected cell from fusing from the neighboring cell.
Right.
But when the virus has to fuse with it, presumably the virus has some of the same tetraspanins as the cell surface.
Exactly.
So the question is, how does the virus able to fuse, right?
Right. And again, it's in principle is somewhat less infectious or less fusogenic. But we just think that it's still enough, or we see that it's still enough, even if you overexpress and thus make the virus incorporate more of the tetraspanins. It doesn't lose completely its fusogenicity. Okay. It just becomes less fusogenic. So again, sort of the idea of a compromise of.
So why do you think that paramyxoviruses have no problem fusing lots of adjacent infected cells, but HIV apparently has evolved a way to prevent this? Do you know anything about paramyxos and tetraspanins? Anybody work on that?
I'm actually trying to think. So paramyxo would be Roberto Cattaneo.
Like measles.
Yeah, measles. No,
mumps.
Actually, we did Roberto Cattaneo.
Yeah.
He contacted us a while ago. Actually, we were looking with him if there was any role and if measles would be. I think he was interested in looking at budding, but somehow it didn't go anywhere. Mm-hmm. So I assume that he didn't really see a role for them.
How do the hantas cause syncytia formation?
I mean, that's a good question, too. I mean, if you think looking in a rodent host that's going to maintain these viruses persistently,
probably not.
That would not be a good thing, but certainly you can get fusion to happen in culture if you drop pH. It's all pH dependent. So maybe it's just a pH phenomenon that
or maybe tetraspanins are involved.
Or maybe. We find them in virions.
Oh, yeah?
Yeah.
And I think, boils all down to environment. I think that's really going to be key. I mean, like when you look at my website, I think that the other thing that I point out is that I think many of us, including ourselves, we are still using systems that are somewhat limited in the sense that we're looking at monocultures of cells, essentially. Mm-hmm. But we know now from whatever work from Mina Bissell or so in cancer research, so that cells can behave completely differently if they're put into the context of a 3D whatever environment with neighboring cells that influence things. And there's no reason to assume that viruses are any different. So I think the context is really critical. And actually, in that regard, I don't know if you saw that paper maybe 3 months ago or so in Nature, a nice study where they put HIV in humanized mice and followed the trafficking of the infected cells. And what they found essentially is sort of like, I guess, support for, because what they found is that these infected cells sort of migrate around and then contact other cells, move on or so, and they correlated that to some extent with spread of the virus in these humanized mice. But what they also showed that a small fraction of these infected cells actually appear to form syncytia. So they saw syncytia, which was sort of not completely unexpected, but still autopsies early on that people have done showed sometimes syncytia in infected late-stage patients or so, and it generally was believed that syncytia would not necessarily would not play a role in the spread of the virus, and perhaps only under some circumstances, like if there's actually a very nice work by David Soll in Iowa, he showed that syncytia sort of can squeeze through endothelial cells, and so his idea was that syncytia sometimes can help infected cells to overcome hurdles or barriers, and so.
I noticed on your website you also mentioned that you're looking at tetraspanins and influenza virus.
Yeah.
So Megan Shaw has found that CD81, which is a tetraspanin, positively regulates flu.
Yeah.
So if you knock it down, you get less flu production. Is that the idea?
Right. Yeah, so that actually this was a very nice. So we started to collaborate with Megan maybe 2 years ago or so, after she published a paper that directly contradicted what we had published a year before. Then it was actually really nice because so we in 2007 I think we published a paper. We were starting to look at influenza virus as a control virus for HIV, and with the idea going back actually to for me one of the best papers ever. David Sabatini, there was a JCB paper in 1983, where they showed a fundamental difference between viruses that are transmitted at cell-cell junctions versus viruses that are released at free surfaces. And obviously, influenza virus was sort of like a prototypical virus that was released at free surfaces, and VSVG was released at cell-cell junctions. He showed that with epithelial cells before they formed monolayers or so. And so we all thought, well, okay, so HIV is a cell-cell transmitted virus, flu goes free surfaces, so they may likely use different platforms. And so we were doing experiments where we treated HeLa cells exactly like use them as controls with influenza virus, and sure enough didn't really see much tetraspanins where we saw the emergence of influenza virus. But then like a year later, Megan Shaw in PLOS Pathogen did a nice proteomic analysis with purified virus and showed, oh no, there is tetraspanins, quite a bit of it actually in flu. So we contacted her, and nicely enough she gave us material, and when we looked at her purified virus, sure enough we saw things also. And it turns out that like when we discussed things with her, it was really just the technical differences that did not allow us to see tetraspanins are there. And then again a year later or so, she had a Nature paper where they did the RNAi screen and showed that CD81 is required or is one of the positive factors for influenza virus. This is great that you find someone who contradicts and then you collaborate with them. This is perfect. That's the way it should work, right?
That's how he does work.
So you're learning this from him, I guess, right?
Yeah.
That's great.
Yeah.
Because that's the way science should be. But unfortunately, not everyone does it that way. Not everyone is open and collaborative. But it's actually good for listeners to hear firsthand an example of that. And that's a good way to end this, actually, because we can't do any better than that. This episode of TWiV will be, as usual, on iTunes and at twiv.tv. And if you like TWiV, the best thing you can do to help us for now is to go over to iTunes and just leave a comment or a rating there. So that keeps us very prominently on the front page of the Science and Medicine podcast directory. If you have any questions and comments, we love to get them. Send them to twiv at twiv.tv. And both my guests today are at the University of Vermont in Burlington. Jason, Botten, thank you so much.
You're welcome. Thanks for having us. Yeah. Pleasure hearing. Looking forward to more work from you. And Markus Thali, my host here, thank you so much for doing this.
Thank you very much, Vincent, for coming here.
My pleasure. Good to hear a perspective from Virology in Vermont. Hey, maybe that'll be the name of this episode. What do you think? VV, Virology in Vermont. I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.